Fortunate to be collaborating with her in some of the congenital heart disease projects, and she's doing amazing work. And what she's going to do or talk today about is infant cerebral blood flow dynamics and emergence of brain function and behavior. Minhui has got a lot of talents and has been really working hard on some of this advanced imaging analysis. So we're really looking forward to hearing what you have to say and meeting with you after too. So thank you so much for coming.
Thanks. So good morning, everyone. I will begin by express my gratitude to Dr. Grant, giving me the opportunity to present my work here. And also it's an honor to be here and to present my work in front of these distinguished colleagues and experts of the fetal and neonatal neuroimaging. So today I'm going to talk about my quite recent work for the infant cerebral blood flow dynamic underlying the emergence of the brain function and the infant behavior development. I can wait for a few minutes.
Come sit down. There's a couple chairs. There's two more up here and one over here. And we can grab some from the room next door too. Some chairs.
So all life activity requires energy. So we can across this lifespan, we can clearly see the energy expenditure rise during the early infancy and the childhood. And this rapid increase of the energy demands mainly to fuel the energetic, expensive organs such as the brain. And in adult, despite the brain is only present 2% of the total body weight, it uses more than 20% of the total body energy. But this number is much higher in infants. It got short. It's used 40% in infant. So this high energy consumption actually is quite important for the underlying maturation process during this early infancy, including the synaptogenesis, dendritic uprising in the cortex, and also the myelination across the whole brain. And although the human brain development is a continual process after birth and throughout the adulthood, but it's most dramatic in the first 2 years of life. Or I should say postnatally, because I know some of the colleagues here are doing the fetal development, and for the first 3 trimesters, the fetal development also quite rapid. So, for example, from the conventional structure MRI, we can see remarkable morphological changes in the brain development. And after birth, the whole brain volume quickly increase and reaching to 80 to 90% of the adult volume by age 2. And besides these morphological changes, we can also use other measures such as keyholes like sarcopy to see the cortical folding pattern quickly change during this time as well. And this overall brain size change actually is come from the underlying gray matter expansion and the white matter maturation. And such white matter microstructure maturation can use techniques such as the diffusion tensor imaging to quantify the underlying microstructure integrity changes. For example, use the fractional anisotropy, we can see the white matter integrity changes during the first 2 years of life. Besides that, we can also use the diffusion chromatography to study the emergence of this white matter tract during the first 2 years of life. And all these developmental changes required energy and nutrition. And the human brain exclusively use oxygen and the glucose for energy production, and the cerebral blood flow deliver this oxygen and glucose to different brain regions for the energy reproduction. And previous study already showed there's a tightly coupling between the regional cerebral blood flow and also the regional cerebral metabolic rate of glucose and oxygen at the resting state. So this makes the CBF, it's a quite good surrogate measure of the brain cerebral metabolic rate. And the CBF dynamic during the adolescent and the adulthood has been quite well studied. However, during the young childhood, especially for the first 2 years life from the 0 to 2, is much less studied, and there's a knowledge gap how this CBF dynamic changes during this time.
That's a good question.
Yeah, go ahead.
So, I mean, this is great, but it seems there's quite a bit of variance, right? Is the variance due to actual people, like children, having different CBF, or is it the measurement variance?
You mean this one?
Yes,
Yeah, I think this is an individual variance.
Okay,
Yeah.
so individuals have this much variance.
Yeah, each dot represents one subject.
Okay, so we think the CBF measurements are actually quite accurate in these.
Yeah.
Okay, thanks.
But you still can see the overall trend from the years.
Yes.
Actually, this quite match quite well with the energy for this one. For example, in the early 10, like at the 9 years old, reach the peak of the energy consumption and reduce until the 21 years old, and then keep constant until 60 years old and slowly reduce. And this match quite well with the CBF finding as well.
Okay.
Yeah.
Great, thank you.
Can I ask another question? For that measurement, did you use the non-invasive SL technique to measure the CBF, or are you using the contrast agent like the DSC to measure the CBF?
I use arterial spin labeling. That's the common part now. Yeah. But one quick way to do is using the non-invasive phase contrast imaging. For example, we can estimate the flow flux at the 4 major feeding artery, including the bilateral internal carotid artery and the bilateral vertebral artery. So based on this phase contrast imaging, we can estimate the global CBF. It's a quick way to do it and quite reliable. And using a large sample size of around 120 infants from 0 to 2 years of age, we can quickly see the global CBF actually rise rapidly from birth to 28 months of age and follow a logarithmic fashion. And for the 1 month, we can see the global CBF around 17 milliliter 100 gram tissue per minute. But at 24 months, it's around 70. So it increased more than 4 times increase. So this is like a very clearly demonstrate the CBF increase quite dramatically during infancy. But since this is global CBF is from phase contrast, there's no regional information. And that's why the next step we were trying to do is, hey, how can we get the regional information for the CBF? And we are using arterial spin labeling in this study.
And can I ask a question about the last slide?
Yeah.
Back to owner's question, how much do you think the variance in that is due to, say, not exactly perpendicular to the vessel or slight errors in the area of the vessel? Do you think some of the variance in the data is from that as well as?
Oh, this is a very good question. Since the velocity is quite different during this time, so even with the phase contrast MI, you have to optimize your parameter. For example, for the first 6 months, and for the in this phase contrast MI, you have a velocity encoding that parameters, and you might want to significantly reduce compared to the adult one. And the vertebral artery actually have a much lower velocity compared to the ICA, so you want to reduce that one as well to make more accurate assessment. And for this one, we use 4 phase contrast MI, which means for each vessel we have one separate acquisition. And for each one, we are trying to, after we do the angiogram, we trying to find out the best location perpendicular to place the phase contrast MI to get the best images.
And for the temporal resolution, because you've got the whole cardiac cycle, right? So you've got the change. How do you get the mean velocity, or how do you calculate the velocity? And do you try to get it at, so you're capturing that full cardiac rate?
Well, I think the easy way to do is you acquire multiple slides. or quite a longer time, then you can average them across this time period. Yeah. So for the next step, we did using the arterial spin labeling, a non-invasive perfusion MRI without any contrast agent, to answer your question, which is quite suitable to study the CBF in the infant and neonate population. However, unlike adult ASL, applying this technique in neonates and infant, there are several challenges. The first one is related to extremely slowed blood velocity. So you need to wait, which means you have a longer arterial transit time. You need, after you label the blood, you need to wait much longer time for the blood to reach the imaging slab to perfuse inside the brain tissue. Another challenging comes from the small brain size for this neonate and infant. For this small brain size population, we required high resolution to achieve comparable anatomic information in adult, which means this is at the cost of a reduced SNR. And ASL is already quite low SNR technique. There's a lot of challenges if you adopt the adult parameter in ASL will never work in this neonate and infant population. And around like 7 and 8 years ago, we optimized like a pseudo-continuous ASL Picasso sequence for use in a neonate brain and reach moderate reproducibility for this neonate CBF and also moderate resolution. And many colleagues in the field already start using the ASL to study the brain perfusion in neonate and infant in different disease conditions, including the congenital heart disease, like ischemic stroke or HIE. Yeah. With this background, today I'm going to talk about the infant perfusion, and it covers four important parts. The first one is we optimize the cutting-edge Picasso sequence to achieve the high-resolution infant CBF map. And with this high-resolution map, we were able to delineate the infant CBF dynamic from 0 to 24 months, and also study the association between this CBF dynamic and the infant behavior development, as well as the functional network emergence during these first 2 years of life. Okay, the first part, I'm going to show how optimization is important, is the key to obtaining the reliable and high-resolution CBF in this infant population. First of all, I always like to, I think, and many of you might agree, for any successful infant MRI scan, one will need an amazing frontline team. That's why for some of the HPC study, besides this protocol setup, different sequence, a lot of time we are discussing the logistic and how to scan the young population. And without this frontline team, it's not possible for me to present the work in the later slides. Yeah, I would like to acknowledge my frontline team as well. Yeah. We start using a 3D multi-shot stack of spiral Picasso sequence to optimize this sequence. And this was developed in one of our collaborators, Dr. John Detro's lab. And this spiral acquisition actually is quite SNR efficient readout, and also allowed us to use the multi-shot acquisition to reduce the blurring and also without the too much sacrifice of the SNR. And with the background suppression, we can achieve, and we use 4-shot here, 4-shot acquisition. And with background suppression, we can achieve the isotropic 2.5 millimeter resolution for this infant ASL.
How did you optimize the duration and labeling delay?
Yeah, very similar like our neonate work, we do a systematic testing for, ranging from 16 to 2000.
So did you do that for each age group? Because, the CBF is going up over the age that you're looking at.
Ideally, it will be great to do in each small age group.
Yeah. And these are all neonates, so the ones that you did here, is that correct?
Yeah, neonate is ongoing one. Neonate is a different situation. After 1 month, actually, the trend is much smaller compared to the 0 to 1 month, the neonatal period. Yeah.
I'm also curious about the T1 difference between adult and also the infant. So the
When you
infant,
say T1 is blood T1 or the tissue T1?
tissue and tissue T1.
Tissue T1.
Yeah.
Okay. Yeah, there are many ways can use to tackle this problem. You either acquire a separate scan. For example, you use collars to acquire the T1 relaxation time of the whole brain, but it will increase your total acquisition time. Another one you can, actually our sequence cover a small module to estimate the T1 already. So for the tissue T1. And another way, if you don't have this acquisition, one way you can do is based on previous literature. There are many literature already established the gray matter and white matter T1. So based on that, you can fit the age-dependent curve. For example, I think Sean Dioni has a lot of study related to this part. Then based on the age, you can fit the gray matter and white matter T1 value, then fit inside your estimation. Then it will get you much better value.
So I'm gonna ask you about the blood T1 too, because you've got the hematocrit changing.
Yeah.
Over that time period as well too. Now, luckily you don't have inpatients, these are outpatients, so you don't have to deal with transfusions and all sorts of other things that can change blood T1, but how do you manage the blood T1 and?
Well, if it's disease study, actually it's much easier because most of them will acquire the HCT right value.
Yeah.
And that one is actually related to the T1. You have an equation can estimate the exact T1. So it's much easier. But for this cohort, we are doing the typical developing children. We are not draw the blood to estimate the HCT value. But this is very similar as the labeling efficiency when you need to input these parameters when you are estimate the CBF. These are all global information. We can use the phase contrast MI to normalize, to calibrate this regional information. For example, the phase contrast MI actually is quite reliable already for estimate the global CBF. But for the regional one, once we estimate from the ASL, then we can do a whole brain average and make these two values the same. Then you use the phase contrast to calibrate the regional CBF. Then this will largely minimize the effect of the global parameters which you are assuming, for example, the labeling efficiency and also the blood T1. This will not be that big problem for the estimation. Yeah. With this optimized protocol, we are not only can get the high resolution and high SNR CBF map, but also excellent text and retest reproducibility of this infant CBF map. This compared to my 7 and 8 years old, that neonate CBF, the reproducibility actually is quite nice. And this is a 17 months infant, and we can clearly see the nicely correspondence between these two repetitions of the scan with the final optimized protocols.
Are you using any motion mitigation strategies?
Prospective, no. But for the motion, but we will also do the motion correction. And from the motion, since we have multiple dynamic, we can estimate the in-scanner motion into one motion parameters. And this one will be used as a variant in all the following developmental curve or the correlation with the behavior and the functional network.
So you don't necessarily correct for it or do any motion correction on the images, but you get a variance measure to add in your statistical analysis?
Yes.
Okay.
Yes. And for the scan with large motion, for example, larger than 0.5 millimeter, that main relative displacement, we will just exclude those large volume, like scrubbing the motion, and also exclude the subject with such high motion in the analysis. Yeah. But in general, it's fine for the infants since they are sleep quite nicely. But I think once you go to toddler, that will be a huge problem. At that time, another way to further improve is adding like the navigator T1. We can also add the navigator inside this spiral Picasso, since we have a quite long time for the labeling that time. After labeling, we have a waiting for some time, and that period we can have a quick acquisition as well. Yeah, I think John's team is already trying to incorporate this VNAV inside the Picasso. Since Dylan is also work with John, and Dylan was developed the VNAV for the structure T1, T2 imaging, and it's quite easy for him to plug this small module inside the ASL. Yeah, this will be extremely beneficial since we are using the multi-shot acquisition. So 4 shot, which means we need to acquire 4 TR to get 1 image. So it will be very benefit to have a motion correction module inside. Yeah. So with the high resolution optimized protocol, we can see compared to a conventional protocol using a 2D multi-slice API, our infant dedicated protocol can achieve a higher resolution with like densely acquired axial slice with much better gray and white matter contrast. But more interesting is we capture more than 3 times more cortical vessel acquired. This is quite important for the brain with such a small size.
I have another question. So for adult, we usually like plan our, labeling plan like 84 like millimeter below the ACPC. I'm curious for the infant, how you plan your.
Yeah, that's based on Han-Zhang's paper, right? The adult below the 84. That's there are many ways can do. Some people say, hey, you can base on the TOF. Then you can get the artery and you will find the best location usually. But another way, easy way is just use the anatomic location as a guidance, like between the C2 and C3. I personally like the C2 and C3 location. I'm telling you a reason, because when MI tech are playing this, they might not get familiar with the like vessel location. This will have individual MI tech like a variance if you play using the TOF or angiogram for this labeling location. But C2 and C3 is so easy. That's why for my acquisition, I usually just suggest them to just use the simple one, C2 and C3. Yeah.
May I ask one question? Do you need a B0 correction for your spiral? Like deep learning?
This one we didn't.
Yeah, okay.
Yeah.
Because I saw like it's like distortion free, like compared to EPI.
Yes.
Okay. Okay, but you didn't consider B0.
We haven't. Yeah.
Okay.
And this one, this is the individual CBF map from the milestone age like 2 and 8 and 17 months across the infancy. And we can clearly see there's a rapid increase of the CBF value. And this is the only individual map. And this CBF distribution actually is heterogeneous across at a specific age as well. But this is much easier to appreciate if we map them to their cortical surface. For example, at the 2 months infant, we can clearly see and the CBF value is higher in the primary region, including the sensorimotor and the auditory region. It's quite clear. But at a later age, we can see the high value are more in the association region, including the prefrontal and parietal lobe. Yeah. And
Often when you're doing ASL2, you'll get, because of the delay from the posterior circulation, slightly slower. Sometimes you get less labeling. I'm curious if you ever saw that variance, at least in our clinical population, we often see the occipital lobes aren't as perfused because of delay differences between the anterior circulation and the posterior circulation. Did you see that in any of your data, or do you think that your labeling duration and so on, your optimization mitigated that?
As long as your post-labeling delay longer than the longest time of your the arterial transit time, it should be able to handle the slow the slow arterial transit time from the occipital lobe. Yeah, but if we can do systematically, I think the ideal way is we can do the multi-delay one, and then from multi-delay one we can estimate the actual the arterial transit time.
Yeah.
Then based on that we can set our PLD. Then this will solve the problem. But this will cost time because you don't want to acquire another separate like sequence for the multi-delay. Yeah.
Question, so is there a group map or individual map?
Oh, this is an individual map. That's why only with this high resolution you can see the detail for the individual variance.
How's the intersubject variability
In the subject variance.
for the same age?
I did not estimate the standard deviation from this map, but I do have a population average one across different milestone age in the latest slide. Yeah.
Do you see some asymmetry around 1 year of age because of there's a handedness and language development? Because here it looks like more or less symmetric.
That's a very good question. Actually, there are some studies saying right hemisphere actually have higher CBF value, but I didn't do a systematic test.
Right hemisphere.
Yeah,
Because you also have right hemisphere somewhat higher.
yeah, but I didn't do the systematic test for the.
Ipsilateral to right hand, which is strange.
How much of the individual variation do you think is driven by different reaction to being in the scanner itself?
When you mean different reaction,
For example, how awake the infant is.
all of these actually is during natural sleep.
Okay.
Yeah. So,
But REM sleep versus non-REM sleep, do you think?
well, that will be difficult too. But I always believe the developmental change we capture is much higher than the REM sleep or non-REM sleep, this variance. Yeah.
The acquisition time to generate a map.
7 minutes. Yeah. Ideal time. Yeah. What if the baby wake up, then there's no data? Yeah. And we for the perfusion we put at the last for the acquisition, so it's quite difficult for me to get a large cohort of the data.
What's your success rate?
For perfusion?
Yeah.
Because before that we have a quite long scan. It's almost the end of the 1 hour, so
Yeah.
I didn't get too much
Yeah.
sequence. Yeah. But I think this HBC, this infrastructure, and this like wisdom from the bigger group, I believe we probably can boost the success rate for the future scans.
Yeah.
Yeah. Oh, this reminds me very quite sometimes study. This data is the PET pioneering study. The images are quite before I was born. Yeah, this study. So the pattern is quite nicely matched the CBF actually. If you take a look at the 2-month one or before 2 months, the high cerebral metabolic rate of glucose are located in the primary sensory motor region or the subcortical region or the auditory region before 2 months old. And later at 7 or 8 months old, then the frontal lobe starting using more glucose, and there's a clear increase for this for the chat. And at 1 year, the distribution pattern is completely different from the early 10. And the CPF actually matched this pattern quite nicely, and it's non-invasive. Yeah, I was wondering, there are much less past study in this maybe it's not even possible for this typical.
You can't do that anymore
Yeah.
because radiation exposure.
Exactly.
Yeah.
So CBF provides us another way to take a look at the perfusion distribution. And with this optimized sequence in hand, the next thing I was trying to delineate the CBF dynamic across the entire the first 2 years of life. And with densely sampled age across the 0 to 28 months, and this is we create the population areas, the CBF map. I did not estimate the variance between this one, but it will be interesting to estimate that and probably to add another value. And in the future, this can use as a normative reference. For example, for the incoming subject, we can estimate how much it deviates from the central line and calculate the z-score to maybe this can use as a informative biomarker.
How many subjects per each age group went into the average?
In total, I have 76 subjects for this map. I think around maybe 7 to 8. So it's still not enough, but it's a good start to take a look at the CBF changes. Since I noticed that all these big consortium parameter is DHCP, BCP, or HBCD, including HBCD, there's no CBF information inside. We have been keeping ignoring the perfusion part from the MI acquisition. Yeah.
It'd be interesting to see how many you need to get it to stabilize, in terms of the variance start to stabilize. Because at least when we're doing the atlases for the ADC maps, around 8 or 9, and it's starting to stabilize.
Oh, that's quite nice.
Yeah.
Yeah. Probably that's my next step to estimate the individual variance. And we can use the adult one as a basis to see how many this will not change that much after some time, and to see, I expect for the infant one, we might need slightly more subject compared to the adult population.
Your white arrow is pointing.
Oh, sorry. So for specific age range, or we can clearly see the CBF distribution is also heterogeneous. So for example, for this younger age range, we can see high CBF value in the subcortical region and also the primary sensory motor region. Precentral and postcentral region actually is quite bright. But this can easily see from the cortical map. As you can see here, this light blue indicate the higher value. Yeah, I can see that. At the younger age group, including the sensory motor and the auditory and the visual region, they have the higher CBF value during this time. And we can clearly see the distribution change across this milestone age. And at a later age, the CBF value is higher in the association region, including the precentral, including the frontal and the parietal lobe. Yeah. And I think this movie also shows the 4D spatiotemporal CBF dynamic, and can also see the switch, the distribution switch actually happen like this age, 6 to 9 months, and the distribution from the high value at the primary cortex slowly change to the association cortex. Yeah. And with this map, we further study the age-related change, how this CBF change during infancy, and we can see the age effect unevenly distribute across the cortical region. And we can see the highest age effect happen at the frontal lobe and the parietal lobe. And using a predefined functional network, and we observe that the frontal parietal network and deformal network actually shows the greatest CBF increase during this time. Although a substantial increase also found in the sensory motor network and the limbic network, but to a lesser degree. Using a data-driven clustering approach, we also reproduce this the different network pattern, and we observe. We find there are 3 clusters based on the CBF increase pattern, including the limbic cluster, sensory motor cluster, and the frontal parietal cluster. This is a completely data-driven approach without using any predefined function network. And this is the first time we observe such physiological hierarchy along the limbic, sensory motor, and association gradient throughout our infancy. Although there are many studies in like a later age, like adolescent and adulthood, they always talk about the association, sensory motor and association gradient. SA-axis, you might already read a lot of paper related to that, but this pattern might not be there at birth or during infancy. It's more, we believe it's more slowly from the limbic sensory motor and slowly emergent to the SA gradient, which is later people talk about in the adolescent and the adulthood. Yeah. And within this cluster, we can clearly see the different CBF increase pattern. And there's a biphasic pattern of the CBF increase, and the red line indicate the average CBF value within this cluster. And we can see the CBF's rapid increase in the first phase and slowly increase in the second phase. And more interestingly, from this segmented biphasic regression analysis, we can estimate the breakpoint age. And the higher hierarchy, for example, the frontal parietal lobe has a higher hierarchy. It has a older breakpoint ages. But for the lower hierarchy, the limbic one has a younger breakpoint ages based on this segmented analysis. And this analysis was reproduced using a Wuerzel-Weiss one. So the one shown here is the cluster level analysis. We also perform a Wuerzel-Weiss analysis. For example, for each Wuerzel within this cluster, we will do a segmented analysis, and based on that, we will have a breakpoint age. And this is the Wuerzel-Weiss, the breakpoint age profile, and we can see the limbic one has a younger breakpoint age, and the frontal parietal one has a much older breakpoint age.
I'm just curious for the relative CBF. Did you match using phase contrast to get the global CBF, or the global CBF comes from like the ASL itself?
For my first slides, the global CBF actually come from phase contrast MI.
So for here you're using the global CBF from phase contrast.
So for all these regional CBF, we also use phase contrast estimated global CBF to calibrate the regional one. So this will make the CBF regional CBF map more accurate. So because a lot of time people are talking about like labeling efficiency will influence your estimation, and the T1 block will influence your estimation, and you have a different age. But with this phase contrast calibration, such effect actually can largely minimize. Yeah. And similarly, we observe the CBF increase rate is much higher before the 6 months of age. You can see this is the milliliter per 100 gram per minute per month. This is based on months, this CBF increase rate. It's consistent with other imaging measures, including the brain volume and also the microstructure measures we can. Based on the like traditional DTI measure, the FA change actually is quite rapid in the first years of life. Yeah. And when we map the CBF increase rate, like a range then toward based on the identified gradient, we can clearly see the gradient has a higher, if the gradient has a higher hierarchical level, the CBF rate with a higher value will last longer compared to the lower hierarchical level. So this further support the physiology hierarchy along this limbic, sensorimotor, and association region. And this is, as I mentioned, this is consistent with other imaging modality observation. For example, this is very old study in our lab, and that time my mentor was using the diffusion chromatography to study the white matter emergence. And the limbic fiber, which is the first row, actually emerged quite early compared to the association fiber, which is showing at the bottom. And the association fiber actually matured like in the late third trimester and myelinate more in the first 2 years of life.
Sorry, I interrupted. One question. The heat map, what is the ordering of the rows? Or is it each one
Each
a subject or?
also,
Oh, each row is.
oh, each row,
Mhm.
each row, for each row is a Warsaw.
It's a mean voxel in that region over your population.
So this is the increase rate. So for each Warsaw, you will have, based on the developmental curve,
Yeah.
you can do the derivative, then you can get the increase rate at specific age.
Yeah, but each voxel has, say, the 8 subjects of that age, right?
All these, so since the trajectory is based on the total 76 subject, so based on that curve, you can derive the increase rate, and then you can get the increase rate map. And
But each voxel is not an individual.
no,
right?
it's not
Yeah.
individual. Yeah, it's a population based.
I was just wondering how you chose the ordering
Yeah.
within each, just curious.
So for this part, it's just based on, for example, I already have the whole brain separate into 3 clusters. Then each Warsaw, I just have a 1, 2, 3 label. Then I just arrange that. Yeah, but we think that specific cluster, it doesn't really matter. But you can still see the pattern, like for the frontal parietal cluster, you can have a longer time period of the higher increase rate. Yeah, and this one is quite consistent with other measurement. We did a similar study using the structure connectivity measure, and we observed the similar increase rate is so obvious in the first 6 months compared to the later 2 years of life. Yeah.
You define the voxel correspondence across subject, maybe, to get that map, right?
Yeah. So this is just an increase rate map, meaning, okay, I think this will be easier. So for this kind of curve are based on 76 subjects, but each curve indicate one Warsaw, which means I can do a derivative, right? The first derivative to get the increase rate for this curve. And at each 10 point, I can get the increase rate based on the derivative.
So something I wasn't clear on, so this is a longitudinal study and what was the time points?
This is the cross-sectional study.
Cross-sectional.
Yeah.
Oh, okay, so this is a longitudinal curve made up of cross-sectional
This is an
data.
age-dependent
Okay.
curve based on
Yeah.
cross-sectional data set.
Yeah,
Yeah.
and then you register each age to the next age, so you clearly registered them.
Yeah,
Yeah,
to a
through
template.
surface template, right?
Yeah.
So then you got correspondence, I think, on the
Exactly.
voxel. Okay.
Yes. That's why we have the increase rate map and arranged based on their cluster and location. Yeah.
The voxel size stays the same, right? Voxel size stays the same?
Warsaw slice.
Size,
Warsaw size. Yes, they're the same.
but the brain size increases, right? So doesn't the number of voxels increase? How do you fix for that?
That's a very good question. This will relate to the partial warning effect. Although the high resolution CBF map is exactly trying to tackle that problem to reduce the partial warning effect. But even with this 2.5 millimeter isotropic resolution, you might still have that. But another thing can kind of minimize that is we haven't did that yet, but it's similar like our previous cortical microstructure analysis to only doing the skeleton value. We think that cortical ribbon, that part can like significantly reduce the partial warning effect, but was not adopted in this analysis. Yeah.
So to be able to make the comparison, are you using not just the registration, but also interpolation? Are you using any interpolation to match the 3-month-old brain to the like 8-month-old brain?
Well, since all these individual brain map will register to a template, you will do some kind of interpretation during this registration process.
Sorry, then what the age for the template? Do you register them to the midpoint or all to the baseline 3 months?
Midpoint. Yeah. And we have an infant atlas developed in the lab, like 1 month, 12 months and the 24 months. So we usually use the 12-month one to match every subject to the midpoint. But right now we are developing the age-specific template, including the 3 months, 6 months, 12 months, and 24 months, including the cortical surface. Then this can be a very, very good resources for the researcher to use. Given the HBCD, this kind of big cohort data will release early next year, and this kind of template and surface, including atlas, are quite needed to study a different imaging measures.
So yeah, I'm not the expert for neuroanatomy, but I would assume there are some miscorrespondency. So for some cortical structures, we might not observe them as early as 3 months. If your template like 12 months, there might be more delicate structures. So would that be a problem when you register them by the surface-based registration?
Oh, this is still volume-based registration. The registration right now, we are still doing volume-based. And after we get the volume data, we map the result to the surface.
Yeah.
Non-linear. For non-linear, yeah. But for the anatomic, for example, the structure, I think during the third trimester, we can see some structure disappear.
Yeah.
But for the infancy, the structure are there, it just grows maybe non-linearly.
And the general topology would be subtly different is kind of the biggest thing probably you'd see, or differential growth of the frontal lobes versus other.
Exactly.
So so is your registration non-linear? Because if you do it linear, it won't match, and some areas it will the others.
So first we need to do a fine linear, then we apply a LDDMM, the non-linear one, to match the individual to the template space.
And you can tell right there because all the surfaces look the same. So that's the surface of a.
That's the surface.
Yeah, of the 12-month.
Yeah. Okay, the next one with this CBAF dynamic review. The next one we're trying to study how the CBAF dynamic actually relate to the infant behavior development. Since the first 2 years of life, besides the brain development, the behavior also change a lot. From we don't talk and then we suddenly talk, we don't work and we suddenly work. So we use to measure this behavior, milestone behavior, we use the Bailey scale to measure the developmental functioning in the motor scale, language, and cognitive. And we observe that along with this RCBF, these are come from the same subject. And we find that the higher CBF value actually is correlate with better skill from this Bailey assessment. And in this analysis, we specifically regress of the age, sex, and another important factor is the family socioeconomic status actually, and other covariant, including the motion from the RCBF estimation. And we can find the different muscles of RCBF significantly correlate with the Bailey scale across different domains. Yeah. And we further link this significant RCBF and behavior correlation to the identified cortical gradient, the physiological gradient. And we find that for the brain vessel, for the significant vessels, we categorize them into different different cluster. And we find out the RCBF with significant correlation with cognitive function are actually primarily found in the frontal and parietal clusters. And CBF with significant correlation with the language are primarily found in the sensory motor the cluster. However, the motor, we find both. Yeah. This also this could be related to the Bailey. It's not that specific. Many of these scores are kind of overlap when you are doing the assessment. And using another behavior test, this is more like the parent questionnaire one, violent adaptive behavior, we find a similar pattern with higher CBF value associated with a better adaptive behavior skills across multiple domains.
I can't see this very well. So your Bailey and Violin are done at the same age or at the time of their CBF measurement?
On the same day.
On the same day.
Yeah, so usually, I think Dr. Grant knows this, when we run HPCD scans, HPCD study, very, very similar, we will do so many components, including the assessment, behavior assessment, or EEG, or MRI. Some of the components will be like combined on the same day. So for our study, we usually do the neurodevelopmental assessment first, which is the Bailey, and the mom will also, or the dad will also fill out this parent questionnaire, which is the Violin. After that, then the baby will go to the scanner.
And they're tired after all that testing.
Yeah, exactly. That's the trick. you want to use this like a Bailey scale, like Bailey assessment to tire the baby, then they can sleep better during the MRI scan. Yeah.
So what's the youngest age for like the baby to have the Bailey?
3 months.
3 months?
Yeah, 2 months, 3 months. Or the assessment is like quite quick for those younger baby, might be less than 30 minutes. But for because not a lot of like behavior has been emergence, right? So it's quite quick. But for the 2 years old, I think that assessment lasts 1 hour, more than 1 hour. Yeah.
And all of your babies their scores are within normal range.
I think so. It's not below 1.5 standard deviation of the normal range. Since this study is we're trying to study the typical development. So one of the exclusion criteria is, hey, if we doing the assessment of their behavior and the score actually is below the 1.5, their standard deviation, and that subject will be excluded. But for the analysis part, you might want to include it to see what's the effect of your brain measurement and how this is related to the brain behavior development as well. Yeah.
So do you think that if you see this connection between some regions and some specific domains, that at some point you can look at the MRI and predict the score at this domain, like vice versa?
Using MRI to predict this domain?
Because if you see it, then you can predict how good is the child.
It's doable, but predict the current 10 point, I don't see the point. Because you can do the behavior at a current 10 point. You don't have to predict, for example, at 3 months. But if you predict a future, like I'm doing the infant scan at 3 months, and predict their behavior at 2 years or 3 years, that will be more meaningful to me. And we have done some research like this using the cortical microstructure at birth that encoding a lot of cellular molecular process. And we use that information to predict the Bailey score at 2 years old. And it's quite reliable. Yeah. And similarly for this CBF, I believe we can also develop model, like machine learning model to predict that. But I'm more interested to predict the future outcome. That will be more meaningful. But especially for example, the autism, they don't get their clinical diagnosis until 2 or 3 based on the behavior assessment. But what if we can do the early prediction? So this will change the paradigm.
But in these models, I think it's a different thing to predict normal outcome with normal data. To predict abnormal outcome, and you don't know if you can do that yet because you don't know whether things are going to have the same relationships or be tied as closely together. So that's still to be determined, right?
Yeah, but from the prediction perspective, I think you want to include in the abnormal one to make your model more
Yes,
robust.
agreed. That's why I'm sort of pointing out, you have to have the abnormal in the
Yes.
model to be able to predict not just normal, but abnormal.
Yes, we had an ongoing study related to the HIE.
Yeah.
And we kind of merged the normal and the HIE data together. And actually the produce much nicer because you have a wide range.
Yeah.
Not only for the brain imaging, the feature variability, because you want the feature variability, but this variability cannot come from like a noise.
Yeah.
It should not be the imaging noise. It should be the individual variability. And then for the behavior part, you also want to cover quite wide range of this outcome. So the prediction will be much better.
For your sample, do you have any follow-up of these children? Like some of them still normal, but probably some of them are some delay after your study, like 6 months later or?
It could be, but so for this, we have a cross-sectional cohort and longitudinal cohort. And I didn't match them with the later age 10 point. So at some point, might be after 2, they may have developmental delay, but
Some of them might.
yeah, it could be.
If
Yeah.
the sample is large enough, maybe you can get some children.
I think that's the beauty of those huge, large consortium like 7,000 kids to have. Then we can have a nice individual variance captured. Yeah. But for our one, maybe quite difficult to get so much sample. Yeah. Okay, so let's move to the final part of the talk. This part I'm trying to link the CBF dynamic to the functional network emergence. Since in addition to these rapid physiology changes and also the structural changes during the first 2 years of life, the infancy is also marked as like a very rapid period of functional emergence. And such functional emergence and functional connectivity change can reliably estimated using the resting state FMI. And for the network hub, which is the interconnected hub region of each network and kind of facilitating the neural signal or communication within the brain. And one can also use the data-driven approach, including the independent component analysis to derive the important network hub, for example, the deformal network and visual and the sensory motor network. And this is using the ICA to derive, and we only use the 1 year to 2 year old infant. We don't include the one before 1 year because we think that some of the function network actually hasn't been emergence yet. If you put the data together, you might not be able to get the deformal network ROIs as well. And using the FMI, resting state FMI, we already observed for the primary regions, primary network, including the sensory motor and visual network, are growing quite rapidly during the 3rd trimester, and the network are well established at 10-bone baby. But however, another vital function network, the deformal network, is not established by the at birth yet, and it's more established quite later, much later infancy. And so let's take a look at how the diamond network emerges during this So here we use the seat-based functional connectivity analysis, and the posterior cingulate cortex is marked by this dash area are seat, was used as a seat to study the connectivity within the network, within the deformal network. At birth, we see the deformal network is not that mature, which means there's not much connection between the PCC and other deformal network hub, including the MPFC, like the LTC or the IPL. However, the brain growth, and then we can clearly see the connections between these regions to the PCC are getting stronger and stronger during these first 2 years of life. And then we map the PCC related functional connectivity to these milestone ages. As we can see here, 0 months, there's not much connection with other deformal network hub. But at 24 months, the deformal network actually is quite obvious to see, and compared to the adult one as well. Yeah. Similarly, we use ICA data-driven approach to get the deformal network hub, also the visual network hub and sensory-motor network hub. And the functional connectivity within this network, we think the deformal network is significant increase during the first 2 years of life. And the FC, we think the visual network, or we think the seminosensory network, there's not such a significant increase, indicate the network is quite stable already, or probably mature quite early, before maybe at birth or before birth. Yeah.
That's a question. So the part or the default mode on the top row, is that the correlation with voxels within that region with each other?
Yeah.
Okay, and so you get that strong a signal at 0 months in that.
Yeah, because you use these pieces as a C, so
Yeah.
you average the both signal in this region, and you can do the correlation with the whole brain, and we can clearly see.
I'm just surprised that within that region, it doesn't increase over time as well too. It looks pretty stable there.
Yeah, pretty stable. This is what we find.
Yeah.
It's pretty stable. It didn't
Yeah.
change that much. Yeah. Then the next one, we try to check the CBF changes within this network region, and we find that for these 3 network regions, the CBF is significantly increase with age. But however, the CBF increase rate is much higher in the deformal network regions, and the dashed line indicate the sensory motor network, just as a reference here. Not only the total deformal network, but also each small deformal network hub has a higher CBF increase rate compared to the sensory motor network. And the next, we try to understand whether this CBF dynamic underline the emergence of the deformal network by performing analysis called the Wuerzel-Weiss coupling between the whole brain CBF. This is the whole brain CBF Wuerzel across subject, and for the functional within a specific network, you will just only have one vector. And for this one, we can do the correlation between the FC within a specific network and the whole brain CBF Wuerzels. Yeah. For example, if we take a look at the coupling between the RCBF and the DMN FC, and we can clearly see the strongest correlation were found only in the deformal network regions, which is marked by the red dash area, not in those sensory motor region or the visual region. And from this radar plot, we can also clearly see the brain Wurzel with significant correlation between the RCBF and the intra-DMN network FC, only much higher in the deformal network region compared to the visual and the sensory network region. Yeah. This correlation only found in the Warsaw specific in those deformal network region. And we conduct similar analysis between the coupling of the whole brain RCBF and the intravisual network and the intrasensory motor network. And actually, as expected, we didn't find any significant correlation. Yeah, so which further indicate the CBF coupling were very specific to the deformal network region in this in the infancy, because this is the time the deformal network regions emerges quite rapidly, but the visual and the sensory motor is quite stable during this time. Yeah. And based on those findings, we hypothesize there's a neural mechanism supporting such coupling of the CBF and the functional connectivity. So for when the CBF increase, there will be more like oxygen or glucose delivered to the different brain region. It will increase the cell molecule and the glucose metabolic rate, and this will increase the synaptogenesis and the synaptic efficiency, and which will increase the both signal synchronization. And based on the Herban root, the neurons that fire together and wire together, eventually the functional connectivity within that specific network will increase. That's the hypothesis we are trying to explain the coupling. Yeah. In summary, we optimize the advanced sequence to get the high resolution CBF map during the infancy, and we can see the CBF distribution are non-uniform across the brain region at a specific age range. And the CBF increased quite rapidly during this time and heterogeneous across the cortical region, most prominent in the heteromotor association cortex than the unimotor cortex. And we also review a physiologic hierarchy along the limbic sensory motor and association cortical gradient through this infancy. And this provides insight for the neurophysical basis for the infant function network emergence and the infant brain, behavior development. And this high resolution CBF map throughout the infancy can also use as a normative CBF atlas for future research, including the disease one, like the HIE or the congenital disease.